Okay, so if we talk about the aerobic respiration, so in aerobic respiration, we are having the glycolysis cycle, as well as you can say that the link reaction, Krebs cycle, as well as electron transport chain. Okay, so these are the mechanism of the aerobic respiration in the form that basically deal with the energy in the form of the ATP. Okay, so here the basically the glucose is broken down. Okay, so if we talk about the aerobic respiration, so we have discussed already in the glycolysis too. So today we are having the next topic we are having, that is link reaction. Link reaction, we are also calling link reaction as the oxidation of the pyroids. Let's see how. Okay, so what is link reaction? This is basically a reaction where the oxidation and decarboxylation occur occur okay, so de oxidation and decarboxylation. So if there is the oxidation, we are calling that as a dehydrogenation too. Okay, so this cycle basically occur in the mitochondria matrix, but after that we are having one step that basically occur in the cytoplasm. Okay, so we are calling that generally as it occur into the mitochondrial matrix, which means fluid we are having in mitochondria. Okay, so start with the cycle. So we are having, as you guys all know, from glucose we are having the two pyruvate. So we are, I'm writing, you can say that directly, two pyruvate from the glucose, which means two pyruvate means three carbon. One pyruvate is equal to the three carbon, so we are having two pyruvate, which means six carbon. So this process I'm talking about, that will be occur in the cytoplasm or cytosol. Okay, so 2 pyruvate will convert into the acetyl aldehyde, which is basically the aldehyde. This is basically the 2 carbon molecule. So here the enzyme that is used, which is pyruvate decarboxylase, is basically used for the conversion of the acetyl aldehyde. With that, there is a product is formed, which is 2 carbon molecules, because as you know that here we are having two pyruvates. So the two, if there will be the one pyruvate, but there will be not, but I'm giving you example, if there is one pyruvate, so the one carbon dioxide molecule will form, but here we are having two pyruvates, so two carbon dioxide molecules are formed. Okay, after that this acetyl aldehyde will convert into the acetyl. How? In acetylaldehyde the oxidation will occur. How? From this acetylaldehyde there will be the NADH will be like formed in the NADH, means that there is the reduction is basically occur into this, and here the oxidation is occur because this one has given the hydrogen to this and reduced into the acetyl. Okay, so here the process with this is called, this is the dehydrogenation, which means that the here is the gaining of the hydrogen also. Okay, so after that this is basically the this process till, you can say that acetyl or till aldehyde, acetylaldehyde, there will be occur in the cytoplasm. Okay, so we are, that's why we are calling this a oxidation and dehydrogenation, right? So we are, that's why we are calling this a oxidation and decarboxylation. We are that calling as a dehydrogenation too of the oxidation. In oxidation we are calling that as a, you can say that dehydrogenation too, and plus the decarboxylation. Here the decarboxylation is also occurred. Okay, so this acetyl is formed. Okay, this is oxidation, this is here the reduction is occurred. After that here the two molecule of the NADH will be formed. Okay, so this will basically move into the matrix of mitochondria. Here the reaction will occur in the mitochondria. So this acetyl will move to the mitochondria with the help of the active transport, with the help of the energy use, and it is will be formed into the coenzyme, this is basically what? This is basically the coenzyme A, and it will form the acetyl coenzyme. So acetyl will convert to the acetyl coenzyme. What is acetyl coenzyme? So this acetyl coenzyme, that is basically coenzyme of A. Okay, so this coenzyme is basically will be, now it is, this is still the ring reaction. The next video we will discuss about the next station, that is Krebs cycle. So there this coenzyme, acetyl coenzyme will be used. So final product we are having, that is acetyl coenzyme. Let me make the structure of this through which you can easily understand. So this is basically a 5 carbon sugar. Okay, in first carbon, in, you can say that in fifth carbon, not one, not one, 1, 2, 3 and 4. Okay, and here fifth. On fifth carbon, as you guys all know, we are having phosphate, and first carbon we are having the, you can say, nitrogenous base, that is here the adenine. Okay, so here if we have, so we are calling that as a adenine monophosphate, but here I have attached one more phosphate, so we are calling that as a adenine. Here we are having phosphate on fifth one carbon, and next one we are having on the third one carbon. So we can say that here is the nitrogen base adenine. So we can say that adenine 3 5 bisphosphate, because we are having two phosphate. One is on third one, and second one is the fifth one. So we can say that 3 5 bisphosphate phosphate. Okay. After that, with phosphate we are having a coenzyme that we are calling that as a phosphopentothermic acid. This is basically the vitamin B5, we are calling vitamin B5. As well as here with that we are having the sulfur is also attached. This is basically the reactive part, we are having the sulfur. Okay, so this was the structure of the, this one, acetyl coenzyme. So the final product we are having, that is the acetyl coenzyme, that will use in the Krebs cycle. Then I will tell you that what it is in actually. So through that cycle you can easily understand. So this was about the link reaction that we have discussed today.